It's like enough.
All right, good afternoon. Let's get started. All right, so I hate to be the bearer of bad news, but your first exam is kind of on the horizon, right? So not next Tuesday, but the week after that. Oh, getting a little feedback here. Let me see if I can do that. Okay. So we still got, a week and a half, a little bit more than that. Just kind of, if you haven't done any homework yet, this weekend is a good time to do that. We are hopefully we'll be through chapter 3 today. So that's kind of a big chapter, lots of structures and terminology there. So put that on your to-do list. As we get closer, I'll announce exactly how much material is going to be covered by that exam. Right now, we're a little bit behind. I'm not too worried about that. We'll just kind of adjust the material there. We'll probably bleed a little bit into Thursday's lecture as to what will be covered on that exam. Again, following Tuesday, not upcoming Tuesday, but the week after that. My exams, you'll be doing them on Scantron, which I know is not great considering the limited desks that we have in here. We'll have to do the best we can. All multiple choice. So as you're thinking about that, I will try to give you some practice questions in class next week, as well as I will post some. I don't post an entire practice exam, but I will give you kind of a smattering of questions from previous exams. So you kind of will have a feel what that will look like, hopefully. So, all right, so last time we finished our taxonomy, we were talking about Carl Loos and his phylogenetic tree. That's why we correctly have 3 domains now above kingdom in terms of taxa. And remember, he used ribosomal RNA to determine that. There were 3 distinct groups when you looked at sequences there. And we started talking about bacterial structures. So we were working from the outside of the cell in, glycocalyx, which we more often would refer to as the bacterial capsule, and flagella. We spent some time on that. So then we started talking about the cell wall. So a little bit about kind of what happens when it grows, what is peptidoglycan, and now we're finally going to get to what is the difference between a gram-positive cell and a gram-negative cell. Okay, it doesn't have anything to do with the charge of the cell. All bacterial cell walls are going to have a slight negative charge, and that is important when kind of you think about what dyes to use in order to see it under the microscope. But it has to do with differences in that cell wall structure. So you have 2 major groups of bacteria, again, the gram-positive and the gram-negative. So the gram-positive bacterial cell walls, what's unique about them? They have lots of peptidoglycan. So remember, that's that polymer, alternating sugars, NAM and NAG with the amino acid cross bridges. Gram-positive cell walls are really not that complicated. They have a lot of peptidoglycan, about 40, somewhere between 40 and 80% of the cell wall dry weight is peptidoglycan. So it's a major constituent of the gram-positive cell wall. And a little mnemonic that I came across, gram-positives, thick, lots of peptidoglycan, gram-negative cell wall, thin, much less peptidoglycan. So the so what of that is that when you're doing a gram stain, which if you're in lab, you're going to do that next week. I kind of wrote that over to the side here. You don't need to memorize that for me in lecture. If you're in lab, you're probably going to need to memorize that. But just kind of do that. We add a primary dye that's purple. Okay, so you add that stain to the cells, and then you add iodine actually that acts as a mordant, and that means it just really pounds that primary dye into the cell wall. We briefly decolorize it with acetone dissolved in alcohol. So because the gram negatives don't have very much peptidoglycan, they get very quickly decolorized. Okay, and then we add another stain so that we can actually see them, which is a different color. It's red than what that primary stain was, purple. Okay, so the gram positives, they hold on to this primary dye. They don't get discolorized very quickly because they have so much peptidoglycan to hold on to that. Okay, and then the gram negatives, less peptidoglycan. So that's the main difference in why that stain stains some organisms red and some organisms purple. Okay, so the gram positives are going to stain purple, and the gram negatives are going to again take up the color of that counter stain, which happens to be red. Okay, what else is unique about that gram positive cell wall? And we'll get to the picture of this in a minute. But they have teichoic acid and lipoteichoic acid. Okay, those are complicated what macromolecule those actually are. They're actually considered sugars. Unique, only gram positives have them. We think that their role has something to do with regulating cell division and also determining the shape or the morphology of the cell, because we know that when we engineer cells that have mutations in that teichoic acid, they are a different shape than what they normally are. So it has, some kind of function there. Let's see. So if we look at your book's figure of the cell wall of a gram positive cell, this fluffy pink stuff, that's the peptidoglycan. I'm just gonna